Welcome. Thank you all for coming here this afternoon to the Mind Visiting Day. As for many of you, you've been here for a number of years, have come here for this event. Others of you are new. And what we do here in this building is that 17 years ago we opened it with the idea in mind that we wanted to accelerate our research in finding cures for neurodegenerative disease. And we thought that if we just go after one disease or another disease, that we would be missing all the possibilities where they overlap. And they have a lot of common features to them. So in opening this building, we took all the walls out, made the laboratories open. All the students have to talk to each other. The faculty has to talk to each other. The fellows have to talk to each other. And actually what's great is they did. And they all started collaborating. And so people started to notice that, as you'll hear, in some of these diseases, there's a second disease. And that second disease may kind of surprise you. So we'll hear about that this morning. It's now totally under the leadership of Merit Cudkowicz and Brad Hyman and Rudy Tansey, who are the directors of Mind. And I get to just come here and blab for a little bit. Okay, so I want you to know that in almost all the diseases, Alzheimer's, Huntington's, Parkinson's, and ALS, there have been, I don't think that's my phone. Okay, so we have actually gone from basic research to an in-man clinical trial, and all of them with new kinds of therapies. And we're continuing to do this more and more rapidly. So it's an exciting time for us. And your support actually makes a huge difference to us. And the reason is that when you're starting out with a couple of really exciting experiments, you may not need very much money to get that, like, does this actually work experiment? And that could be a smallish experiment. You go like, holy cow, you know, it may take you $10,000 or $20,000 to get the supplies and go and look at something completely new. But if it works, then you go, holy cow, we're off to the races to get a new way of looking at it or a new therapy. So today, you're going to hear from a series of people. First is going to be Brad Dickerson, and he'll tell you about his work on memory disorders. And then we'll hear from Mark Albers, who studies both memory disorders and ALS, so that's going to be interesting. And then Todd Herrington is going to talk to us about all the ways that we can actually go in and machine our brain and change the way we behave with different kinds of deep brain stimulation and other things. So that's very exciting. And then finally, we have Nutan, Sharma, and Chris Bragg. We've never presented on dystonia here, but today we are going to, because there's some very exciting findings that I think you'll all enjoy hearing about that have changed our way also about thinking about dystonia. So I'm going to introduce Merit to come and say a few words to you.
Thank you, Anne, and thank all of you for coming today to spend time with us and to hear from our amazing physician scientists about the work they're doing to try to develop new treatments for people with neurodegenerative diseases. The work that takes place here and really with all our collaborators all over the world is really just amazing in neurology right now. We're at a new crossroad where finally we're getting some treatments that are really working for our patients. Just in 2017, we have our first gene therapy for a childhood form of motor neuron disease. We have a new treatment for another rare child disorder called Batten's disease. There's a new drug for ALS that came on the market. There's new treatment for stroke and a new treatment for multiple sclerosis, all within the last 6 months. It's really an amazing time for all brain disorders, and a lot of that research and that collaboration is coming from people here at Mind and at MGH. Just this year also, the first gene therapy trials for Huntington's disease and ALS have started. And that is just something that I don't think 20 years ago or even 10 years ago we imagined was feasible. So it's really an exciting time. And we are seeing a lot of people going into the neurosciences. So we're getting a lot of young talent to help us really make these breakthroughs. So a lot of our breakthroughs here are from that collaborative spirit that Anne, with her leadership, set up here in Mind, and from the philanthropic support from many of you and other of our patients and friends. You'll hear this amazing story from all our speakers, but most recently, really from philanthropy, a major breakthrough in dystonia that you'll hear from Chris and Nutan that we think is going to be rapidly translated into treatment for our patients with that. Having unrestricted dollars is just enormously helpful for our young investigators and our not so young investigators to really allow them to take risks, to think big, and to really go after what they think is going to make a difference. So I want to just thank all of you for that. And today, I want to not blab on too much either, and let us hear from our speakers. So with that, I'll introduce Brad Dickerson, who's one of our leading researchers in memory disorder. Thank you.
Great. Well, it's a pleasure to see all of you here tonight and to have a chance to talk with many of you already this evening. I just want to mention I've been showing this more and more painting by one of our patients who very nicely painted the historic Bullfinch building of Mass General. So I'm going to try to provide a high-level overview of some of the work that we're doing in the memory disorders and frontotemporal disorders programs, and hopefully set the stage for how we think about moving, as Anne and Merritt said, from the lab to the stage where we're giving people medicines or other kinds of interventions to help treat their symptoms when they have one of these different kinds of neurodegenerative diseases. So we always have the patient at the center, and we start with how do we measure symptoms that people are experiencing. And typically what we do is we talk to the patient and family and often examine the patient with a neurological exam or other things that we can do in the office. But we're finding more and more that some of these measures are still relatively crude in terms of what they capture about the patient's experience with the illness or what we're able to potentially measure with some new kinds of technologies. And so we tend to use forms like this. So one of the disorders that falls under the memory disorders rubric is primary progressive aphasia, which is a language problem. And one of the reasons it's been challenging to get clinical trials moving in this field is because most of the scales that have been used so far have been adapted from Alzheimer's disease, where the problem is more one of memory. And so we developed a scale called the Progressive Aphasia Severity Scale that captures different aspects of speech and language and is enabling us to measure in a little bit more fine-grained ways progression of patients. So you can see that you can rate the impairment on a scale like this where 3 is more impaired and 1 is less impaired. And you can see that some patients from baseline, when you first measure their symptoms in the dark blue, stay fairly stable over time. If the light blue is progression, they have a little bit of a change over time. Whereas other patients like this one have a fairly big change over time in these different categories of their language function, so going from mild to moderate severity in a fairly short period of time. And this is one of the mysteries of these illnesses that we still are trying to understand better, is how to make the best prognosis and how to capture some of the variability in people's rates of progression. And there are others of us who are working on using advanced technology. So this is a PhD student that we're working with in the Speech and Hearing Sciences program here, who is quantifying speech. And so you can see million times it's from Mary's mother. That's what the patient is speaking. And here's the acoustic analysis of the waveforms of the sound that you can get from that. And she's been quantifying a variety of aspects of the rate of word production, the pauses that are occurring in between words that people speak. And she's had to do this in a fairly cumbersome way manually, but we're now taking advantage of some of the new voice recognition technologies that are emerging that we all use on our cell phones every day to make this a much more efficient process and something that we could potentially move into a more automated and possibly even remote method of assessment. So these are ways that we're trying to move forward in our ability to measure the kinds of symptoms that people living with these illnesses experience, and that we need to ultimately show are changed by interventions that hopefully improve people's quality of life. These symptoms are coming from changes in the circuits of the brain that we'll hear more about today. So we've gotten increasing ability to use imaging to measure changes in the structure and the functioning of brain circuits, and you'll hear more about that today. But this is just an example of how specific some of these problems can be. So this is all of our, what we presume would be all of our normal brain function when we are speaking and comprehending speech. So it's mostly on the left side. So this is the view as if you're looking at my brain from the left. And this is the view as if you're looking at my brain from the right. And you can see that when we put a person in a fMRI, a functional MRI scanner, and measure the functioning of the brain while they're either reading sentences or responding to cues to speak, there's a lot of activation in different parts of the left side of the brain, the so-called language network, and a little bit in the right side. And in this condition that I'm alluding to called primary progressive aphasia, where people gradually have selective loss of their ability to either speak or comprehend language, the damage, the shrinkage in the brain is in these areas, which is almost identical to the location of the normal network for language function. So why is it that these diseases are sometimes so selective when it comes to causing problems in a certain brain network and sparing many other brain networks? And are there ways that we can harness that specificity to help people compensate for the problems, even if we are still struggling to try to improve their overall disease progression? And then the changes in the circuits in the brain, at least in neurodegenerative diseases, are a result of pathology. And we're now really in a revolution in the field where we have new kinds of imaging and other kinds of biomarkers that I don't have time to get into today, but that measure changes in the molecules of the brain that we ultimately are thinking are responsible for these problems and are trying to target with therapies. And you'll hear more about that from other speakers today. As one example, what we're hoping for, and we're still not here yet, but we've made a huge amount of progress over the last decade and even just the last 5 years in doing what this slide outlines, which is if you have a person that's got symptoms of memory disorders or a dementia, and they might have shrinkage of their frontal lobe, for example, we want to know, one question we have is, does this person have Alzheimer's disease, which is made of amyloid plaques and tau tangles, and we now have the ability to image these molecules in the living person's brain, and that'll ultimately connect with what we hear about from other speakers in terms of what you can measure in the laboratory models of these diseases or in postmortem brain tissue. And so when people have positive amyloid and tau scans, they most likely have underlying Alzheimer's in their brain. If they have negative amyloid and tau scans, they most likely have one of the conditions that's related to ALS or Parkinson's potentially, where there's not so much amyloid or tau at all in the brain, but there are other molecules that are still in the works in terms of our ability to measure them. And in other cases, there are people that don't have amyloid plaques, but they have a lot of tau. And this is where we've made quite a bit of progress just in the last 5 years here, and really have worked with Teresa Gomez-Isla and members of our nuclear medicine department in collaboration to validate some of these new technologies against some of the brain tissue specimens that are in our brain bank here in this building. So really remarkable progress that's already made its way into clinical trials and is beginning to be used to show what drugs do to these molecules in the brain. And you can see that over time in this one person's brain, you can get a sense that the tau started out here, and a year later it progressed a little bit, both in nearby areas that weren't so affected originally and also in more remote areas that are part of the language network in this person's brain. And then I'm just going to briefly go through these last things because some of our other speakers are going to spend more time on them. Laboratory models to understand disease mechanisms are so important, and we're now getting better at being able to connect those laboratory models with what we can measure in living people. You'll hear more about that, but one example of that is with something called stem cell reprogramming, and we can talk more about that offline, or other speakers may touch on this. And then laboratory models to screen treatment. Some of these kinds of technologies allow us to mimic the molecular abnormalities in tau or amyloid or TDP-43 or some of the other things that you hear about from other speakers in the laboratory petri dish, basically, and allow us to put compounds on those neurons that are growing in the laboratory and see what they do, see if they help rescue the problem. And again, I'm not going to spend time on that because we'll hear more about that from other speakers. And then ultimately, we want to take these back into clinical trials of the treatments that we want to give back to the patient and see if it does anything, see if it helps them, see if it changes what's going on in their brain, and ultimately helps their functioning. And there are networks that I had the chance to talk with several people about today that before the session, networks of centers that are collaborating. This is one example. There are many other examples, but this is the Alzheimer's Disease Research Center's program. Our Alzheimer's Disease Research Center here is led by Dr. Hyman and Dr. Gomez-Isla. And these are all the approximately 30 centers around the country that do a huge amount of collaborative work to try to do more than what any of us can do by ourselves. And there are many examples of that. And I'll just close by saying that along with philanthropy, we participate in a lot of advocacy efforts to try to change the regulatory environment and find other incentives to work with partnerships between academic medical centers like this one and industry to really accelerate the progress toward treatments. This is a shot of our frontotemporal disorders unit led by Katie Brandt's efforts to take us to the Capitol for Rare Disease Day every year because many of these diseases are pretty rare, and we need to really get legislators and industry partners' perspectives on those diseases that are not such lucrative targets. And I'll just finish by saying that all of you should be aware that President Obama established the National Alzheimer's Project Act that, for Alzheimer's disease and related disorders anyway, has really done a tremendous service to get our Congress and state governments moving forward to try to change not only research but also care and support and treatment for patients living with these illnesses and accelerate the science that we're able to do. And there's a National Advisory Council, and Dr. Hyman here and Katie Brandt, who, please raise your hand, Katie. People should talk to Dr. Hyman and to Katie Brandt because they're part of the Advisory Council now in advising the priorities for this strategy when it comes to how we're doing this at a national level. Thank you very much for your attention.
Thank you, and thanks to everyone for coming out tonight. This is terrific. It's so great to see so much strong, good energy in the room. And thanks, Brad, for a great overview, and you definitely helped me set it up, so thank you. So I'm going to tell you a story that is not possible except for two things: mind, and the fact that there's no walls here, as Ann alluded to, because you'll see how much collaboration was involved in this story. And secondly, it was not possible without philanthropic dollars, because this was a high-risk effort and something that we would not be able to get funding for initially. And so I just want to thank everyone who's contributed and been involved, and particularly the Wilkins family, who's supported me over the years. And they couldn't be here tonight, but I still want to thank them. It's been terrific. So why have these diseases been so tough to crack. And we've been working hard on them. I think there's three things that we need to take into account to study these. One is aging. These are all diseases of aging. And we need to understand the biology of aging better. So for example, there's mutations in genes, presenilin 1 being one that Rudy Tanzi in this building was one of the ones who discovered it. So we know if someone inherits that gene, you have a 100% chance of getting Alzheimer's disease, and yet it takes 4 to 5 decades to get the disease. So what is happening in those 4 to 5 decades? It's not a pediatric disease. What's happening? We need to understand that, the aging. The second is, and we see this in these diseases, in ALS you can see it, there's an orderly propagation of the disease. So someone may start with leg weakness, and then later they'll develop arm weakness, and we can see it. In the cognitive disorders, we can also see the disease as we follow patients over time move throughout the brain, and it moves in a way that's consistent with neural circuits. And Brad showed evidence of that with tau. You see that how it's moving through the brain. We see the disease move. So there's mechanisms then. So in other words, it's not just random loss of neurons that creates these diseases. You have to take down a circuit. By the time someone comes to see us in the clinic, they've lost 50% of the neurons in that given circuit. So it's not just random loss. There's mechanisms such that when one cell dies, it makes neighboring cells or connected cells more vulnerable. And that's how you take down a circuit. So we need to understand those mechanisms and how to intervene. And the third, and I think this is the most challenging, is that the relevant cells are disappearing. So unlike cancer, where the problem is growing before you and you have tissue to study, in neurodegeneration, the cells are disappearing. And so in this analogy here, what you see is this is a predator, so that's the disease, taking out. And so when we have brains, and thanks to Brad's efforts over the years, there's 1,800 brains upstairs for us to study. And when we study those brains, we're looking at these guys, right? We're looking at the cells that survived. And so we have to be careful because some of those things are going to be survival mechanisms. The changes that we see may be survival mechanisms. These are the cells that are surviving in that noxious environment. And so we need to understand that and see how cells respond in a positive way and a negative way. So the story that I'm going to tell you is related to Alzheimer's disease and ALS. It really starts with, or I think it's based on one of the most seminal observations that we've seen in the last 5 years coming on. This building is from Teresa Gomez Isla, who was here. I think she's here. There she is, yeah. So, and by the way, she is the Ann Young Endowed Professor here, and so she stands on very broad shoulders. And so Teresa has collected a series of cases where people have normal cognition, for other reasons, they've come to autopsy. And surprisingly, the density of amyloid plaques and neurofibrillary tangles is the same as patients with Alzheimer's disease. And so the question is, what's the difference? And so Teresa's done great work to say that there's two things. One, there's neuronal death in the cases of Alzheimer's disease, not the other cases. And the second is it's neuroinflammation. There's inflammatory pathways that are upregulated in these brains that we don't see in those resilient cases. And so we've been focusing on that, trying to understand what is the cause of this inflammatory response in the brain. So the immune system, its job is to distinguish self from non-self. And we think of it in the context of viruses and bacteria fighting those off, right? But as one ages, you can see that you might change. So the immune system, as you age, begins and learns that this is you. Now, as you age, some people change over time. And so that as you change, the immune system begins to recognize parts of you that are maybe not the same. And so, well, the idea is that the aging, you transform from self to non-self. Now, how does this happen in the brain? So here's how we're thinking about it. And we think this is present in some cases of Alzheimer's disease, in some cases of ALS. We don't think this explains everything, and that there's going to be multiple different flavors of these diseases. But the idea is that every neuron in our brain is as old as we are, and every day it takes 1,500 breaks of the DNA. And there's repair mechanisms that fix it. So every cell in the brain, 1,500 times a day, is getting breaks and repaired. Now, just like any other repair process, it's pretty good. It's very good, actually, but not perfect. And so as you age, these cells, as old as we are decades old, they're accumulating these changes. And eventually, the idea is that you start to make things that originally were not part of what the immune system learned is what's yourself. And so you're calling that genomic instability. And from that, you can make then what we are calling here is double-stranded RNA. So that can come from your own genome. We've been able to show that. Of course, then viruses can contribute to that. And over half of our genome is what's called dark matter. It doesn't get expressed, but it has a lot of inverted repeats. And they're normally repressed. As you age, they can get de-repressed, you can start making them, and they have the capacity to form in this. Now, the way we're built is if you see this inside your cell, that is a virus until proven otherwise. Doesn't matter where it came from, that's a virus. And so you activate innate immune signaling, you get a neuroinflammation, and then you get neurodegeneration. That's the idea. Okay. So now, here's a brain from the brain bank upstairs. It was recruited by Merritt or one of our teams, somebody with ALS, and 2 different patients, actually. And this is a technique that we've been working on so that we can now image multiple things on the same brain section. So we can start to ask questions about pathways within the same brain section. But what I want to show you on is here's double-stranded RNA inside the cells, the neurons, in these patients with ALS, but we don't see them in control cells. So we're seeing these viral. Now, in patients that have a particular type of ALS, where it's associated with a gene, C9orf72, we can actually purify these double-stranded RNA and show that it came from the mutated gene. And we're learning in sporadic ALS where we don't understand, there's no gene associated, we're trying to figure out where is it coming from, what's the source of it. But that's the species there, we think the pathogenic species. Now, orderly propagation. This is the model, we're using a mouse model here in my lab upstairs. This is a whole mount of the mouse olfactory system. So this is the nose, inside the nose. Here's the skull, and this is part of the mouse brain. And we have designed and engineered mice that express the double-stranded RNA exclusively in the nose. They don't make it in the brain. And what we see is that those cells, and it's about 1% of the cells making double-stranded RNA. And what we can see is that we lose 40% of the neurons in the nose. So cells that are not making double-stranded RNA are dying. But more importantly, I think in terms of propagation, is that we're seeing cells in the brain dying. So let me show that to you. So just to orient you, so the mouse, the nose, the nostril will be here, the eyes back here. Okay, so, and we're going to take a section right through here. And that's what you're seeing here. So these are 2 brothers. One got the disease gene, one didn't. Here's the nose, and here's the brain. So this is an inflammatory marker. So in the nose, we see no inflammation. We see no inflammation in the brain. In the diseased brother, we see inflammation in the nose, where the double-stranded RNA is expressed, but we also see inflammation in the brain. And if we blow up these 2 regions, we can use the beautiful anatomy, the organization of the olfactory system. And so here's the side, the normal brother, no inflammation. Here, what you're looking at is the nose neurons come in and they connect right here, inside these circles. These are where the second order olfactory neurons sit, and they extend out and the connections occur inside those circles. What's really cool is these cells here are neurons that are not connected to those nose neurons. And yet, they're showing the inflammatory response. So we've got spread from the nose into the brain and beyond. Now, what's cool is that if you appreciate this here, these neurons in the brain are dying. This is the brain itself. So I hope you can appreciate the difference between this olfactory bulb and this olfactory bulb. And if we quantify that, it's about a 15% reduction. So double-stranded RNA in the nose is sufficient to cause neurodegeneration in the brain. So that's the idea. So this can happen now anywhere within the nervous system. And that's the idea that we're... Now, relevant cells are disappearing. That was the mouse model. We're collaborating with a laboratory over at Harvard Medical School. It's Laboratory of Systems Pharmacology. And these are cells that Du Kim, he may be here tonight, he's on the 3rd floor. Du Kim has generated and shared in the collaboration. So these are human neurons that are stained green. They're made green. The double-stranded RNA is tagged with red. Okay, so what you're going to see is this is a movie over 48 hours. Each frame is an hour. And you're going to see that the double-stranded RNA goes into the neurons, and then it comes down, surrounds the cell body, and then the cell's going to die. Okay? So I'm going to turn the movie on, but pay attention to this cell, and then after this one dies, we'll move to in this region. There'll be a second one. Okay? And I hope you can appreciate that it's coming into the cells, going down the processes, and see it's surrounding here, this cell, and now it's gone. And now here's one here, double-stranded RNA around it, and now it's gone. So that's, in 48 hours, we're seeing human neurons in culture dying. And now if we do a dose-response curve of double-stranded RNA, we see that at low doses, we don't see death, and then at higher doses, we start to see death. And that's associated with the same inflammatory response we see in the brains and in spinal cords of ALS. And so our guess is that as we age, this is young, and at some point you start with these mutations, you're growing, and other reasons, it grows, it triggers, reaches a threshold and triggers, and you get death. Now we have an assay. So we can screen drugs and see if we can block inflammation and block neuronal death. And so here's a drug, and there's a class of drugs that some of them are FDA approved, that as we increase the dose of the drugs, they rescue the neuronal death in these human neurons in culture. So we're very excited about trying to understand that in more detail. And on the tour, I'm happy to talk about this in more detail. And so with that, because some of them are able to get into patients relatively quickly, we're very excited about putting together what's called a basket trial. And this is a concept that we borrowed from cancer. So in cancer, if you have an oncogene and it acts up in your lung, you get lung cancer. The same oncogene acts up in your liver, you get liver cancer. So now you have a drug against that oncogene, and we'll take some patients with lung cancer and patients with liver cancer as long as the tumor has the same oncogene. Here, what we're trying to do is we'll take patients that have ALS, as long as they have this inflammatory response, and we'll take patients with Alzheimer's, as long as they have that inflammatory response. We're agnostic to the disease. What we care about is the mechanism. And so we're going to screen those patients. Alzheimer's, ALS, we also see it in reflecting Bragg, in frontal temporal dementia as well. And so we're screening these patients by what's in their spinal fluid. And if they have that inflammatory response, that's what we're trying to, that's the clinical trial that we're designing right now. And that's thanks to both Merritt's help, James Berry, and then also Stephen Arnold on the Alzheimer's side, and everyone working together. And so what are we going to do? So this is my last slide. It's going to be critical to define what are samples in the spinal fluid. And so Teresa and Brad are involved in a study that's been going on, I think, for 38 years. It was started by John Groudon, and Brad's taken over, and Teresa, where patients come in, or subjects come in once a year, and they get analysis, neuropsychological testing, they see a doctor, they get a diagnosis, et cetera. Some of those have volunteered for spinal taps. And so what you're looking at here are people that are cognitively normal, people with subjective cognitive complaints or symptoms. So how's your memory? Not as good as it used to be. Neuropsych testing, completely normal. And then patients with mild cognitive impairment. Memory's not as good, and there's some changes on their neuropsych testing. Not enough to have dementia. And so we're looking at one of these inflammatory markers in these CSF. So in the clinically normal, we're not seeing too much. In some of these patients that have some symptoms but no test results, we're starting to see some that are higher. We have 2 patients here with mild cognitive impairment, one that has an inflammatory marker and one that doesn't. So these are the types of patients that we would like to recruit in the trial. And what's particularly interesting is this is actually the same patient who volunteered for 3 spinal taps. Year 1, year 2, year 3, and they had subjective cognitive symptoms at the time of their spinal taps. That was 2013, 14, 15. They've continued to come in and be seen. 2016, 2017. They're not yet in for 2018, but they now have mild cognitive impairment. So we're it's one patient. We have to be. It's early days, but the idea that we could maybe pick this out and then intervene at that stage and see if it would help. And so there's many people to thank. Merritt and Ann have been extremely supportive over the years. Brad Hyman, I'm in his research unit. He's been involved with that. And Steve and Osley in my lab, Du Kim and Rudy for the cell culture work. On the clinical trial design, Merritt, James Berry, Stephen Arnold, and Matthew Frosch in pathology, and many other people to thank. It's really a collaborative exercise. Thank you for your attention. Happy to answer any questions.
How's everyone doing? A lot of science in a short little period of time here. Hope everyone's doing well. We're gonna talk for the next 10, 15 minutes or so about Parkinson's disease. And I just wanted to start by asking, how many of you know someone with Parkinson's disease? Wow, basically maybe 3/4. It's very common. There's about 1 million or so people in the United States that have Parkinson's, and it's second only to Alzheimer's disease in prevalence among neurodegenerative disorders in neurology. And it touches many of us in our lives. And I'm going to talk about Parkinson's disease from maybe a little bit of a different perspective from what you've read about or thought about or heard about. I'm going to talk about it as an electrical disorder in the brain, as a disorder of brain network activity, right, that emerges not because the whole brain is perturbed, but really because some small parts of the brain are perturbed, and that gives rise to abnormal activity across the whole brain. And I'm going to suggest that there are ways that we can interact with the brain, say through electrical stimulation, that can help to normalize some of that brain activity and improve the lives of people with Parkinson's disease. So first, just a little primer on Parkinson's disease. This is a slab of the brain, a real brain, cut through like this way. Right? And I want to draw your attention to this part at the base of the brain called the midbrain, and in particular, this area called the substantia nigra, the dark substance, which in a normal brain here are these 2 little dark streaks here. These are actually little pigmented neurons that produce a chemical called dopamine. And actually, these little clusters of neurons here deep in the brain make the dopamine for your whole brain. And if you look over here in the Parkinson's brain, you see that the overall structure here looks about the same, but it's missing these dark streaks. And if you were to look under a microscope, you would see the neurons that make dopamine have degenerated, have died, and that means that the brain is starved for dopamine. That's not the only thing that's happening in Parkinson's disease, but it's a really important set of cells that's lost in Parkinson's disease. Well, why is that important? I'm going to now move from this sort of real view of the brain to maybe a more schematic circuit diagram of the brain. I'll tell you this is actually a really simplified model of the brain, but don't get lost in the details. I want to make one very simple point from this. If the healthy brain looks something like this, the brain with early Parkinson's disease really looks something like this. Most of the brain looks fine, but it's lost these for example, dopamine inputs to this circuit. But the loss of that little dopamine input to the circuit gives rise to abnormal activity all throughout this circuit. That's despite the fact that if you were to go to a trained neuropathologist and have them look at, say, some of these other parts of the brain in early Parkinson's disease, they could scarcely distinguish that brain from a healthy brain. Right? Most of the brain is healthy and functioning okay, or could be, but owing to this degeneration in specific parts of the brain, the whole brain network activity is abnormal. And so there's good news in that. Right? It suggests that if there's some way that we could go in and help to normalize this brain activity in the otherwise healthy and functioning parts of the brain, that we could improve patients' symptoms in Parkinson's. And in fact, we do something just like that with a treatment called deep brain stimulation. It's like a pacemaker for the brain. A wire called an electrode is inserted into the brain, into some of these deep structures, attached to a pacemaker-like unit in the chest. And we deliver stimulation to the brain. This is someone with Parkinson's disease treated with deep brain stimulation. In this panel, his DBS is off. In this panel, his DBS is on. You can see that a number of things that DBS is helping with. He has quite a lot of tremor when his DBS is off. You may also appreciate that when he's trying to move his hands, that his movements are not fluid. He's also quite stiff. It's also true for the legs. If you were to feel his torso is also stiffer when his DBS is off. You'll see in a moment that he cannot stand without assistance. He also has a great deal of difficulty when his DBS is off, even initiating a step. It's called freezing of gait. And even taking a single step is a challenge of that therapy. And so for specific movement symptoms in Parkinson's disease, DBS can really be life-changing and transformative. But for those of you, which is many of you we saw who've known someone with Parkinson's, you realize that Parkinson's symptoms aren't static. Right? They fluctuate dramatically from hour to hour over the course of the day. And sometimes people have involuntary movements called dyskinesias. Other times they may have tremor. Other points they're slow and stiff or might have that freezing problem. And the deep brain stimulation that we deliver now is delivered totally ignorant to the state that person is in at that moment. So this thing is just on and stays on in the same kind of setting 24 hours a day. And so one of the things that my lab is working on is to try to understand what the brain activity is like that gives rise to these different kinds of symptoms in Parkinson's disease, and to figure out how to target stimulation therapy to treat those specific brain states, as opposed to delivering this one-size-fits-all kind of stimulation. And this notion of adaptive stimulation is something that heart pacemakers have been doing now for over 15 years. A heart pacemaker records from your heart. If it's beating too slow, it stimulates to speed it up. If it enters an abnormal rhythm, it might shock the heart to get it back into a normal rhythm. Right? We don't do anything like that with deep brain stimulation. And I think in the future, deep brain stimulation could be much more effective if we could understand these target brain states we're trying to treat and deliver stimulation that was targeted at those brain states. And so what are these sort of target brain states? Well, some of them are the kind of symptoms that you saw there that DBS already treats, like tremor or slowness of movement. But also, I think there's promise that DBS could begin to target things that current modes of DBS don't treat very well. Things like freezing of gait, or maybe non-motor symptoms, cognitive and psychiatric symptoms of Parkinson's disease, like impulsivity, cognitive decline, depression, apathy, and others. So what is it going to take to move us from our current approach to DBS to understanding more fully how DBS is interacting with the brain and ultimately allowing us to shape how we deliver stimulation therapy in a more intelligent way? We now have tools to really start to map out the effect of our deep brain stimulation on the whole brain circuit. I showed you before this slab of a real brain, but this is actually taken from someone who's passed, in this slab of brain cut in a pathology lab. But we can now get very similar imaging in people who are alive using MRI scans. And we can take those plots of the anatomy that I showed you before and model actually where those structures are in the brain in 3 dimensions, right? Now an individual person's brain. And we can say, model where the individual person's brain electrodes are relative to those structures. There's some variability from person to person, and so we can begin to say, well, how much, where is the best hotspot, say, for controlling tremor symptoms? But MRI can give us more information than that because we can actually derive from MRI some information about the connectivity of each of these spots in the brain to the other spots in the brain. So we can say, this person's electrodes, how does that region connect up with other parts of the brain? This can all be derived non-invasively using MRI scans in our awake patients, and we can begin to map out now based on this imaging, right, what are the overall brain networks that we think we're engaging with our current DBS, and begin to map which networks we think are critical for the beneficial effects and indeed side effects that we see with DBS. And I was also making electrophysiologic measurements using EEG to map out in individual people which brain networks are activated by their specific brain stimulator. And where are we taking all this? Right? In the last just couple of minutes, I wanted to introduce one little experiment that I think suggests how novel kinds of brain stimulation could treat different kinds of symptoms in Parkinson's than we normally think of DBS as treating. So one problem that can emerge in Parkinson's is something called impulse control disorders. These are compulsive behaviors like compulsive shopping, could be compulsive eating. A common one is compulsive gambling. In some patients, it can be quite severe, even financially ruinous. So how would we study, say, these kinds of behaviors like compulsive gambling in the laboratory? One way is we set up kind of a mini Foxwoods in the laboratory, and people can come in and in a sense, gamble in the laboratory while we record from their brain activity, right? To begin to understand what is going on in the person's brain as they engage in these kind of behaviors. And I was told not to show too much detailed data, so I had just a couple of graphs that I want to show. And what I'm showing here actually is brain activity in a structure deep in the brain called the subthalamic nucleus as a function of time. And then you can see that the activity in this structure was different. It was higher when the person was going to bet low in a specific situation and lower when this person was going to bet high. Right? That was very interesting to us. This is a structure that we know is engaged in more than just movement, but this signal was quite strong. A couple things are interesting about it. One is that if you actually look in time when they place their bet, it's out here. So it actually means that on average we can read out from their brain whether they're going to bet low or bet high about a second or so before they actually place their bet. Which the casinos may be interested in, yeah. But more it suggests that this structure is really engaged in this kind of decision making. And perhaps even that stimulation of this structure could alter gambling kinds of behaviors. And so what this graph is showing is on a particular situation in this gambling game, whether the person is very likely to bet in a risky way and risk a lot, which would be all the way up here, or very likely to make only safe choices and be down here. You can see that if we deliver no stimulation, they tend to be here somewhere in the middle. If we deliver stimulation to the structure that's not really at the time they're betting, at some other time, it doesn't really do very much. But if we deliver stimulation specifically around the time that they have to place their bet, it actually bias the individuals to bet more conservatively, right? Which is what we had predicted from the physiology that we recorded in their brains, actually. And so, this is just a little, nugget of an experiment, right? We're doing a lot of work now following this up, trying to understand how is this happening, what brain networks are we engaging that give rise to this effect. And I want to say that this is not going to be a treatment for impulsivity in Parkinson's disease tomorrow, right? There's a lot of steps that come between the beginning of a basic finding like this and translating it into a clinical therapeutic. But I think just more generally, it highlights how these structures that we're already deriving great clinical benefit from in a disorder like Parkinson's, stimulating it in novel ways may give rise to treatments for other kinds of symptoms that we heretofore haven't been able to treat very well. And more generally, I think this lays out an approach where brain stimulation and other kinds of modulation could treat a broad range of neurologic and maybe even psychiatric conditions. So A couple minutes for questions. People have been kind of shy about questions so far, but I'm really very open for questions if people want to ask them now, or I'm going to stick around afterwards, so happy to take questions then too. Yeah. So let me break in for just a moment. I mean, I think you're making a lot of really important points.
Referring to duodopa.
Yes. Yep. Yeah. And so duodopa is an intestinal infusion of dopamine that can be very helpful for some patients. I think we all agree that the pace of research is always slower than we want it to be. And I think one of the things that's tough is that it does require a lot of work, and you never really know when the next big breakthrough is going to come. And so sometimes you work very hard and you just inch along, and then sometimes you work very hard and things really leap forward. I actually think, so our next speaker is going to talk about a condition, dystonia. I think we'll hear how over really concentrated effort over a very short period of time, they made enormous strides. And I wish that we had that model for all of our diseases, but every day we get up and we try to work and we try to push it forward, and we hope for those big breakthroughs in Parkinson's as well. So, no, it was great.
Oh, you have another question, sorry.
I think I want to be respectful of other speakers' time. I don't know how we are on time. Well, we can do one more, I guess, and then, yeah.
The DBS patients you showed in your presentation, is that strictly an on and off of the electrodes, or were they on other medication as well?
Yeah. So in the fine print there, that person was off of their normal medications, right, which accentuates the effect of DBS between on and off state. Yeah. So there's a much longer discussion to be had about the nuance of how DBS combines with medications, but that video definitely highlighted the maximal on to off state between the two.
Thank you.
Yeah. Good. All right.
All right, so thank you for coming. So I'm going to speak about another movement disorder, which is also a circuit disorder, and which can also be treated with deep brain stimulation. Right? So this disorder is called dystonia. Many people have not heard about it unless they know someone who has it. It's actually the third most common movement disorder, and it's a disorder where the brain is actually too active, and the brain starts sending out too many signals telling muscles in the body to contract. It can affect any part of the body, although in children it tends to affect the arms and the legs more, and in adults it tends to affect structures, the neck and above. So these are some common types of dystonia that people can get, right? So first you can see a woman with dystonia of the neck. Right? Her shoulder is up, her neck is turned. This makes, it can be quite painful, and it makes driving very difficult. So one of the most common problems when adults develop dystonia in the neck, first people think that they've just pulled a muscle, and then they realize that it's persistent and that they're having trouble driving, and that's what brings them in to see their doctor. Dystonia can also affect the foot. That man's ankle is turned in, and this can make people a fall risk. And this is most commonly what you see in children who develop dystonia. And then lastly, people can develop dystonia that only affects a specific, very highly trained task. Musicians can develop dystonia. So you can see that this musician can no longer put his fingers down on the fret. And there are several musicians whose careers have been cut short because of this disorder. So what causes dystonia? Well, it turns out that there can be many causes, but what we have spent a lot of our time working on is identifying genes that cause dystonia. Up to 27 genes have been identified, and one of our colleagues who works in this building has been involved, is the person who has identified 4 of those genes. We also know that you can get dystonia after a stroke or after traumatic brain injury. So we are seeing some forms of dystonia, particularly in soldiers. So I'm going to show you a video of a boy who developed DYT1 dystonia. This is the first mutation that was identified by our colleague, Lori Ozelius. It typically affects children around the age of 12, and it occurs in either a leg or in an arm. So a young boy came to see us who developed dystonia. He had normal development. When he was around 11, he went on vacation with his grandparents. They went hiking out west. And over the course of hiking, the grandparents noticed that when this boy would walk, his arm would go behind him like this. But otherwise, he was fine. He was hiking, he was eating okay. They got back from the trip, and the grandparents just mentioned it to the parents. The boy started school, and then he started having trouble writing. So he had been fine up until that point. He could hold a pen, he could write. Now he was having trouble. And so eventually, over the course of the year, this boy went from just having trouble writing and having trouble controlling his arm to having trouble walking. His trunk was affected. So I have to apologize. I don't have a pre-surgery video of him, and that's why I'm describing it to you. But so his trunk was affected. When he walked, he would go back like this, and his foot would turn in, and his neck would go back. By the end of that school year, he was in a wheelchair. And there was no clear family history, but we tested him for this gene and found that he had it. And deep brain stimulation does work quite well for this. And so here he is the day after he had his battery put in. So you can see that about a week or two earlier, he had had his head shaved so that the leads could be placed in his brain. Because he was a young boy, he actually thought it was cool to get a huge part of his hair, his head shaved. He wasn't worried about the cosmetic effect. So he was feeling a little sick this day because he had gotten his batteries put in the day before, and he was just feeling a little nauseous. Mom is behind him. But you can see that when she removes the support with the pillow, that he starts going back. The muscles of his back will just push him backward, and he's putting his hand behind his head to try to give him some support so that he can keep his head up. You can see the arm starts jerking. The trunk, the muscles in the back are pulling him backward, right? So we actually could not get him to stand up that day. This is him a month later in the office, and now you can still see he's pulling back a little. He needs to put his hand behind his head, but he's able to stand and walk. This is him 2 months later. His back is straight. He doesn't have the need to put his hand behind his head. At this point, I had to write a letter to the school to say that it was okay for him to go back to gym class because the teachers didn't know, what to do now that he was doing so much better. And here he is 3 months after surgery. His shoulder is up a little bit on the right. His left foot turns in just a tiny bit. But, he's riding a bicycle. He was doing all the stuff that kids his age are supposed to do. And this young boy is now all grown up. He's in his late 20s, and he's a pharmacist in New England. So we can only get him to come in now when he realizes that his battery is starting to run out and he needs a replacement, right? So now we're going to talk about another rare form of dystonia that we've begun to study, really thanks to donations. X-linked dystonia Parkinsonism is found in men whose mothers come from one part of the Philippines. So before I began working on this, I had a vague sense of where the Philippines were, but did not know anything about the details of the geography. First, let me show you. Here's Manila, right? And southwest of Manila is the island where these men can trace their maternal ancestry. Okay? And this disorder was first described in the 1970s. Men develop this dystonia when they're in their 30s, so they typically have already had children, and that is how the disease continues to progress. And they have features of both dystonia, we're going to show you a video, and also features of Parkinson's. So you can see some things that are common to the video I showed you before. This gentleman has dystonia of his back, right? He can't sit up straight. But also his mouth is opening uncontrollably, right? His head is turning to the right. So based on all this muscle contraction, he actually needs more calories in a day than someone without dystonia. Yet because he has periods where his jaw is open, he can't get enough calories in, right? And so one of the things that we have seen over time is that these men need more calories, they get less calories in, they become malnourished. Tuberculosis is endemic in this part of the world. They develop respiratory infections, including tuberculosis, and die from a combination of malnutrition and infection. So you can see here, even with support behind him, there's constant motion, constant pulling back. Okay? You can see his right foot is turned in. He is also slow in his walking. He has trouble turning. Right? Someone has to hold on to him. So the balance and difficulty in walking is more similar to what is seen in Parkinson's disease, and he has trouble standing upright without assistance. And now, turn it over to Chris.
So 5 years ago, a group of people came to us and basically said, the world has ignored this disease for 40 years. It primarily affects people on this small island who are impoverished and disenfranchised. And these people had the means to change it and wanted us to take this on in a really big way. And so with their help, we launched what has been one of the biggest initiatives in this department since its inception. And our center is focused exclusively on this disease, not only to support work that we do here at Mass General, but we actually have launched a consortium of labs and clinical groups around the world that we've brought together with funding to work with us on taking this disease on. And it's been a truly awe-inspiring initiative, and we've really, really been honored to be a part of it. What's shown on this slide are some of the many organizations that have applied to work with us and that now form this network of scientists and clinicians. So when we started this, we had no DNA samples to look at. We didn't know what was wrong in this disease. We didn't have cells from these patients to look at under microscopes in dishes. We had to create all of that, and since the vast majority of these patients still live in the Philippines, we had to build this infrastructure for research and clinical care in the Philippines, working with local neurology partners. And so as part of that effort, we've sent teams led by Dr. Sharma to the Philippines to staff a clinic on this island. We work with local neurologists not only to improve that delivery of care, but to collect DNA samples from these patients and to collect skin cells and blood cells that we can grow in the laboratory and study. We construct pedigrees so that we see the family inheritance pattern. In the process, we've worked with scientists in the Philippines National Institute of Health to promote genetic counseling, which has been actually a completely new field in the Philippines, to educate these people about what this disease is and what the risk is of transmitting it to their kids. We have collected brains by working with pathologists based in Manila so that we can study them here and learn what the problem is. And as a result of that, we have literally thrown the kitchen sink and everything we've had at this disease. And in 4 years, we feel very confident that we found what the problem is. We had an army of people at MGH that took this on, and I will spare you the complicated science because I think you've probably heard a lot of that today. But I'll tell you two things. One, we found out what the problem is in their DNA. Patients with this disease have a stretch of junk DNA that's inserted where it shouldn't be. And that actually observation was led by Lori Zelius, who's here in the room. We also found what we think that causes in cells. When we look at these people's cells in a dish, we find that the DNA is turned into RNA, which is the main function of DNA, incorrectly. We don't know that that is the problem that takes place in people and causes their disease. But the thing that is most exciting is there are other diseases where that is the problem, and they're being treated now with technology that didn't exist 5 and 10 years ago. So we've gone in 4 years from knowing almost nothing about this disease to pinpointing the genetic cause and have a leading candidate for the problem that is something that could be fixable. And that to us is absolutely awe-inspiring. And so our initiative now is to build the system to conduct a clinical trial as we continue to understand the basic science more. So I tell people absolutely, steadfastly, who consider supporting medical research here or anywhere else, that philanthropy dollars can really make a difference, because in this disease it is making a difference, and we're very excited to be part of it. So happy to take any questions you have, or we can move on to our tours. So.
Explain what junk DNA is for all these people.
Well, the interesting thing about human beings and other species is that over the millions of years that we've evolved, there have been viruses that have entered us, and viruses that have inserted their DNA into our genomes. And in some cases, that is thought to have been a good thing, that may have actually shaped our evolution as a species. And the vast majority of these remnants of viral DNA littered throughout our genomes is thought to be completely benign and do nothing. But some of these classes of DNA, when parents give birth to children, can move from one location in the genome to another where it shouldn't be. And sometimes those things get stuck in a place that causes a problem. It inserts itself, and what should be together gets broken apart, and bad things happen. And so that is what we think is that is what we found happens in this disease, and we understand a little bit better. So that is an answer to your question.
And are you going to talk about the triplet repeat? I mean, the six.
Okay. If you would like me to, sure. So in addition to what we found.
Just compare it to Huntington's.
Yes. Okay. Okay, I was going to spare you scientific details, but my boss is telling me to give you more. Okay. So we actually found beyond this that not all patients with this disease are the same. So where we found that there's this junk DNA, as I call it, inserted in a place where it shouldn't be, we found that within that is a stretch of DNA that's repeated over and over and over again. And the number of times that it's repeated is not the same among patients. And so some patients have a small stretch of DNA, and some patients have a really long stretch of DNA, which is what happens in a lot of other diseases like Huntington's. So people with the smaller insertions, they do a little bit better. They don't get sick until much later in life. But the people with the really long insertions get sick at younger ages, and they don't do as well. And so this is a common pattern in neurodegenerative disease. We call them repeat expansions, where DNA, for whatever reason, is repeated to variable extents when it shouldn't be. And that causes all sorts of problems. And so that is what specifically we found. Any other figures I should walk them through? Or I'll bring better slides next time. So, thank you very much.
Thank you for coming. It's really a great honor for us to host you here, and I hope you've enjoyed it. Thanks a lot.